Video I'll be saying about what are free radicals and how they are formed, and what are the causes for the formation of free radicals and pathological effects of free radicals, and then the how we remove the free radicals by using antioxidants, types of antioxidants. This all we will be learning in this video. This is a formation of free radical means like this is a normal atom, outermost orbit containing the paired electrons. All these electrons are paired because and there is a loss of one single electron from the paired electron. There is a vacant space here. This is a valency is there. One unpaired electron is there. This is a valency. Unpaired electron, this we got to, in order to satisfy this valency, to try to react with other molecules and destroy them and make them also free radicals. So this is one unpaired electron in the outermost orbit. Any unpaired electrons in the outermost orbit, this is known as a free radical. This is the formation of free radical. The properties of free radical, because they are rapidly destroyed, they are unstable, they are rapidly destroyed by reacting with other molecules. They are autocatalytic. They react with other molecules and convert them also into the free radicals. So they are autocatalytic reaction. Free radical production mainly depends upon the like formation and either due to increased production or defective degradation. There are these excessive free radicals because increased production of free radicals, or they are not degrading properly. Now we consider this as the production and removal of oxygen, reactive oxygen species, and their effects on cell unitary. How these are produced during the inflammation reaction, chemicals or radiation or reperfusion injury. These are the causes for the production of the free radicals. Oxygen enters now. Oxidative phosphorylation takes place in the mitochondria. So oxygen need to be entered into the mitochondria. After oxygen enter into the mitochondria, there will be incomplete reduction. Reduction means, I already told, rig. Rig means gain of electrons. So it is this oxygen gains one electron and forms superoxide. Superoxide with the help of superoxide dismutase, SOD, superoxide dismutase, it is formed it forms hydrogen peroxide. Hydrogen peroxide undergo Fenton reaction with the help of ferrous ion Fe2+ to form hydroxyl ion, hydroxyl free radical. Hydroxyl free radical, which is the most powerful free radical, and all these reactive oxygen species will destroy DNA, destroys protein and the fatty acids and phospholipids present in the cell membrane. These reactive oxygen species modify the proteins, and protein breakdown and misfoldings will be there. The reactive oxygen species damages DNA, and there will be mutations in the DNA, and there will be breaks in the DNA, and fatty acids in the membrane and the plasma organelles all are breakdown. Superoxide reacts with nitric oxide to form peroxynitrate anion. The reactive oxygen species include this superoxide, hydrogen peroxide, hydroxyl ion. These three are the examples for reactive oxygen species: superoxide anion, O2 minus, hydrogen peroxide, H2O2, hydroxyl ion, OH minus, OH, that is one free electron is there. This is all about the causes and pathological effects of and the examples for reactive oxygen species. This is a generation of free radicals, whereas this is a removal of free radicals by using antioxidants. Initially there will be spontaneous decay. They itself get two free radicals can react and then become neutral, or a free radical scavenger system. That is this is not useful. This is this damages this free radicals damages this thing, no. So this is converted into less toxic this thing. Superoxide by acting on superoxide dismutase, this is an enzyme. By acting superoxide dismutase, it is converted into hydrogen peroxide. This hydrogen peroxide further with the help of catalase, in RBC you did experiment in the biochemistry that in catalase test, that is benzidine test. When RBC contains catalase, that breaks hydrogen peroxide into water and nascent oxygen. That is catalase. Or glutathione peroxidase. With the help of glutathione peroxidase, hydrogen peroxide is converted